system, in particular the history of the bioregenerative life support system. So sort of, you could think of it as the prehistory of one of the bioregenerative system we saw earlier today. So these systems, bioregenerative life support systems, were developed in NASA, by NASA in the 1960s. And the idea was, as you know now, that they would provide astronauts with breathable oxygen, nutrient stores, and a source of biological waste removal. And they would do this by using simple organisms, most commonly algae. And algae would grow in the spacecraft and form a series of symbiotic relationships with their human cohabitants. These systems were never put into practice for reasons I'll explain, but the history of their research and design affords occasion to think about the broader cultural structures that condition the possibilities for knowledge at a given historical moment. So I'll talk today about two different kinds of multi-species spaceflight, or bioregenerative spaceflight systems. First will be the single organism system that we used algae, and the second was kind of more radical idea called the multi-species system that would form a complex ecosystem in the space cabin. So when NASA was first established, research on the potential uses of algae in closed systems was well underway. It was part of a broader surge of interest in biology, in algae's mechanisms of photosynthesis during the early 20th century. So as part of this big shift towards studying algae, in the 1940s, the US Air Force and Navy had begun awarding grants to biologists and botanists to develop techniques for culturing algae in closed environments. So the logic here was that by supplying breathable oxygen and decomposing human waste, algae might impel sojourns to the otherwise inhospitable places like the deepest trenches of the ocean or the outer bounds of the atmosphere. So when NASA was first established in October 1958, these algae research initiatives migrated from its aegis, although the Air Force never wholly abandoned its efforts. So NASA's two first flight programs, Mercury and Gemini, those are through the 50s and 60s, those two programs sent astronauts into orbit for short flight durations, between 15 minutes and 3 weeks. And these missions relied on physicochemical systems to sustain astronaut life. So sort of this is a schematic of it. So in this system, water, oxygen, and food were carried on board to last the entirety of the flight. In the future, though, NASA's ambitions included flights of longer durations and more distant places, most notably with a series of manned Mars landings scheduled for the 1980s. And for these, because of weight concerns, it would no longer be feasible to bring full stores of supplies along. So it's here that bioregenerative systems sort of emerged as a viable alternative. So NASA started awarding research grants to botanists and biologists, and these grants yielded several promising algae-based life support instruments. Successful models included the continuous culture chamber, made by a guy named Jack Myers. I'm sorry that...
Try using the keyboard.
Can I go back? Yeah. So these are like the best photos I've been able to find so far, and they're a little, it's hard to tell what's going on. But on the right is the continuous culture chamber, and on the left is I think it's the microtorella, which was produced by two sanitation engineers at UC Berkeley. So there's like a variety of these devices that start popping up in the 60s. And these devices all comprise the same basic units: an algae sample, an aqueous growing solution, and a light source to simulate solar energy, but they targeted different life support system functions. So using a dual bacteria algae system, the microtorella would break down solid human waste and as a byproduct dispense drinkable water. This system, its designers believed, would be relatively straightforward and, quote, highly dependable. Meanwhile, designed to facilitate gas exchange between algae and astronaut, the continuous culture chamber and another one called the recyclostat were quite elaborate instruments. So I don't know if you know much about algae biology, but algae's capacity to synthesize oxygen declines significantly as it grows in density. So a denser culture is inhibited at the bottom from receiving adequate exposure to sunlight, and metabolism rates in general decline significantly as the plants matured. So the idea was to sort of address this and to maximize efficiency. No, don't do that. What?
Wait till now.
Wait an hour.
Okay.
You did okay.
I didn't wait an hour, did I? Okay, well, I'm just gonna keep going. So to maximize efficiency, these devices all sought to maintain algae, quote, at one point on its growth curve, and that's at a bloom state when it's in its most pure and productive state. So achieving this meant that the culture solution would require near constant dilution with fresh growth medium and regular culling to remove mature algae cells and prevent the proliferation of mutants that would contribute to a more variable growth rate. And the device also needed to maintain algae in a state of continuous circulation within the growth tank so that each cell received equal exposure time to the light source. So they really want to sort of make the most pure kind of algae they can. So when it came to sustaining the life of other creatures, these models bore encouraging results. In 1958, Jack Meyer's continuous culture device kept 2 mice in a sealed glass container alive for over a month, and after several tweaks to their systems, other researchers encountered similar results. But scaling up to the human environment meant contending with the device's fundamental design flaw, which was instability. With so many interlocking components necessary to keep the algae in its most productive, pure state, the threat of system malfunction or failure loomed large. So even a cursory list of daily maintenance tasks that would demand an astronaut's attention is maybe useful to think about here. The astronauts' maintenance responsibilities would include regulating growth chamber temperature, culling algae overgrowth, diluting the culture medium, analyzing pH levels and nutrient content, repairing leaks, and monitoring for the accumulation of toxic gases, removing algae foam buildup, changing the light bulbs to maintain consistency in light exposure, feeding the algae with vital nutrients not supplied by human waste products. So you get the idea. So the system, in other words, would evince a kind of watchfulness, as I've thought of it. And this watchfulness was not due to its own fragility, but rather to that of the humans who would depend on it. And that's to say, algae proved unwieldy in its most productive bloom state, but if it were allowed to effloresce unencumbered, it would readily progress to a more mature, but less predictable and more variable state. So the astronaut's long-term survival was tethered to the purity of his life support system, a purity that, in turn, hinged on the astronaut's sustained attention to both the hardware and wetware of the system. As one commentator quibbed, the astronaut's continued existence would rest on his, quote, horticultural efficiency, his skilled attention, and green thumb. So while all this is going on, at the broader scale of NASA more generally, administrative clamor for a bioregenerative system was escalating. By the early 1960s, the United States was, by all accounts, abysmally lagging behind the Soviet Union's space-related achievements. Sputnik, with President Kennedy's 1961 vow to land an astronaut on the moon by the end of the decade, NASA amplified its operations across every aspect of space travel-related research. So in the bioregenerative systems context, NASA continued to award research contracts to botanists and biologists, but they also endeavored to expand these efforts by bringing in a new scientific discipline, and that is ecology. So under the guise of systems ecology, American ecology at this time was in the midst of something of a renaissance, shaped by scientists who sought to distance the field from the more descriptive work of previous generations and reframe it as a science with explanatory power. So accordingly, ecologists moved to conceive the planet less in terms of species diversity or biogeography, which are categories that emphasize difference, and instead in terms of the universal system dynamics that underpinned even the most seemingly disparate terrestrial environments, the dynamics that, it was believed, maintained ecosystems in a state of steady-state equilibrium. So from these efforts to formulate a new kind of ecosystem ecology, emerged a distinct understanding of the ontology of the natural world as one defined by the interplay of complexity and stability. The idea was the more complex a system, the more mechanisms it developed to maintain itself in equilibrium, the less susceptible it was to perturbations, and the more readily it could recalibrate back to its stable state. So at the forefront of this turn to ecological systems were brothers Howard and Eugene Odum. And their commitment to this explanatory power of the relationship between stability and complexity is pretty difficult to overstate. In the 1950s, at the behest of the U.S. Atomic Energy Commission, the brothers conducted a survey of a coral reef in Inuitak Atoll, which was one of several sites in the South Pacific where the U.S. was executing its nuclear test program after World War II. So they were sent to Inuitak to do a big survey of the coral reef after it had been exposed to a nuclear bomb. Actually several. So considered against the backdrop of the crippling destruction generated by the atomic bombs at Hiroshima and Nagasaki, the ODUM's findings were striking. In the wake of a multi-megaton thermonuclear explosion, the reef had largely recovered, appearing within a year, about the same as it had before the blast. So for the ODUMs, this phenomenon of self-restoration was evidence of the profound invincibility of the reef's steady-state stability. So over the course of millions of years, they concluded the region had clearly established a series of, quote, self-regulating interactions that yielded armaments with which to return to its pre-exposure state. So this is a quote that is really, I think, emblematic of their commitment to this idea of steady-state balance. So it's this link between stability and complexity that laid the foundations for both ODUM's bioregenerative life support systems designs. And it also informed their trenchant critique of the algae approach. That the algae approach, structured around a single organism, was unstable came as no revelation to them. And that has gotten turned around. So you should just know that that's a cool photo from the archive. And what it says is, so from what we know about our own biosphere life system, Eugene Odum explained in a grant proposal, it's evident that no one species is ever, quote, reliable unless it's functioning as part of an ecosystem containing other symbiotic organisms. So in other words, the system was not diverse enough to establish the mechanisms necessary for maintaining stability. So instead, the ODUMs proposed the multi-species life support system, a system in which a variety of organisms, all known to coexist in the same ecosystem, would be reassembled and reorganized into the spaceship environment to form what they argued would be an integrated self-maintaining system. These organisms would be selected not for any particular species-specific traits, but instead for their functional role in the ecosystem. Their role as metabolizers of waste, producers of energy, consumers of detritus, etc. So the key to designing these systems then lay in identifying, quote, the minimum number of functional ecosystems, functional components, and the minimum diversity needed for stability in an ecosystem. So their method would be one of trial and error. Introduce men into a closed system of 2.5 acres per man, along with multiple compatible species, continue the seeding pressure until a self-stabilized, fully competitive system begins to be sustained. Eventually, they continued, biogeochemical circuits would become organized by selection, and the constant pressure of maintaining a man present would provide a return circuit for him as part of a new climax ecosystem for space. So in mimicking environments known to have existed in the natural world for millions of years in a steady state equilibrium, the multi-species system would demand little by way of inputs or maintenance on the part of the astronaut. The stability of this equilibrium, when translated into the space cabin setting, would intrinsically confer reliability. So in fact, so confident were the ODUMs in the soundness of this design plan that they eschewed the need to understand the component parts of the system in exact numericized terms. They argued, in an ecosystem of which man is only one, this role of one species does not need to be so precise. And in fact, quantification would in many ways be impossible. No one element in the system could truly be, quote, tested, since, as they put it, the success of the organism depended as much on the size and diversity of its biological environment as on its internal physiology. So NASA administrators were less than charmed by this argument that you wouldn't need to quantify every element, as you might imagine. So they ultimately denied Howard's grant proposal, and after Eugene's 3-year run in the 1960s, they declined to renew his, so they never scaled up beyond simple microcosm prototypes. So it is worth noting, though, that these design proposals were deeply influential to scientists who were involved in the design of Biosphere 2. So perhaps understandably, in NASA's view, the proposals had displaced the astronaut and his well-being to such an extent that the system became one in which it was, quote, immaterial as to which species gains ascendancy. The Odom system of complexity was interpreted as one concerned only with maintaining of, quote, some life in some balance. So, in the end, neither life support system, the multi-species, nor the algae, was ever incorporated into NASA space cabin designs. The multi-species system was quickly deemed untenable and never progressed beyond early experimentation phase, and confidence in the viability of the algae-based system quietly waned as concerns about the system's overall reliability continued to mount. And by the 1970s, NASA's support for algae research had winnowed to just a few lingering contracts, and you may have noticed that NASA is still funding a lot of research on the sort of agricultural aspects, but less so on the bioregenerative in terms of life support system. So these were never realized, but I think this history is still worth knowing because it reminds us that the logics behind these technologies have histories in politics. So reading these histories in politics against the grain affords occasion to recognize the ways that the ways the story unfolded was not at all inevitable, and in fact, it could have been otherwise. Thank you.
Thank you very much. So, one question?
So it's really interesting to think about this idea that more complexity is equaling more stability. And I think for me that really, because my deep background is in engineering, where kind of stability comes through redundancy. And so you want like a simple enough system that you can make it redundant and therefore it will be stable. So first is just the comment of I wonder if redundancy ever comes in this conversation. However, what this got me more thinking about as your talk progressed, and this is going to get confusing I think, and is back to this idea of modeling, is so what you're talking about at heart is like these bioregenerative systems, which are of course fundamentally different from the mechanical systems, which kind of require this idea of redundancy and imagination of redundancy versus the imagination of maybe complexity and stability behind the bioregenerative systems. And yet the bioregenerative systems, as I understand it kind of from your talk and looking at the slides, relies on a fundamentally mechanical imagination of the system as cybernetic. So I kind of wonder, we always think about these like coming together of the biologist's imagination, the engineer's imagination, and why does it never quite work? And I'm wondering if it has something to do with this like fundamental not agreeing with the role of complexity in the system. So I don't know, those are just like these little pieces that I was seeing throughout your talk, and I don't know quite what the question is, but sorry, that's the worst.
Well, I like two responses come to mind. First, I think that often the Odoms in their framing of these, in their sort of proposals, complexity and redundancy are conflated. Like the idea that you'll have, because you don't need to quantify, because if one part, you don't need to know which exact or precise organisms are carrying out which function, because it's imagine that there'll be multiple. So like complex, and that leads me to think more about how maybe biology and engineering have this sort of impasse at various points, but ecology, especially as the Odoms are configuring it, is engineering. It's really like it becomes
it's using, like the engineering model and systems to apply it to not quite an engineering system in some way. Like I'm just wondering if there's like a model conflict. Maybe there's not. I don't know.
For them, no, but I think that maybe that's what ultimately becomes the unpalatable factor for most other observers, most other people.
Yeah.
Let's talk more.
Okay, thank you